Hello and welcome to Nikolay's genetics lessons, and today I got a question from one of my subscribers, and here's a problem. The sequence that cell 1 recognizes and cuts is as follows, and since its recognition sequence is 6 base pairs long, cell 1 will on average cut once every 4 in the power of 6 bases, so that gives us an average fragment size of 4096 base pairs. However, this method of calculating average fragment size does not take into consideration that GC content is 38%. How do I calculate the average fragment size that cell 1 will produce while taking into account the CG content and AT content percentages? So, first of all, how we got this fragment size of 4096 base pairs. Basically, once again, we have 6 base pairs, so let each empty box here would represent each nucleotide. So 4, 5, and 6. And basically we just can say that for first place we have 4 bases to choose from. It can be adenine, guanine, cytosine, and thymine, and 4 bases to fill the second place, third, fourth, fifth, and sixth. And now when we multiply all these numbers, we are going to get 4096 bases. Or we also can say that probability that in the first place we will find guanine would be 1/4. Probability that in second place we will find thymine would be 1/4, and then probability that we are going to find cytosine would be 1/4, guanine also would be 1/4, and adenine 1/4, and also cytosine would be 1/4. So, and our answer would be 1 out of 4096 base pairs would be probability that if we will take a random sequence of 6 bases, that sequence would be as follows. So now let's do our calculations, taking into account that GC content is not like we usually do in our calculations, where we suppose that guanine always equals to cytosine and thymine always equals to adenine, and we suppose that each base pairs would be 25% according to these calculations. But this time we see that GC content is 38%. So if we know that say GC equals 38%, we instantly know that TA content would be 62%. And we also would instantly know that guanine would make 19% and cytosine would make another 19%, and we also would know that thymine would make 31% and adenine also would make 31%. So now let's use these numbers in our calculations. Instead of using percentages, I'm going to use numbers on the scale between 0 and 1. So 19% would be 0.19, and 31% would be 0.31. So what is the probability that the first place would be guanine? And we know that this is 0.19. What is the probability that the second place would be thymine? And we know that this is 0.31 probability. Cytosine next also would be 0.19. Guanine 0.19. Next adenine 0.31, and the last one, cytosine, would be 0.19. Now we just have to multiply all these probabilities, and we are going to find probability that we will take a random sequence of 6 base pairs, and the sequence would be guanine, thymine, cytosine, guanine, adenine, and cytosine in this particular order. And what else? This is going to be just a probability which would equal to 0.001252338. So this is going to be again probability that if we will take 6 base pairs randomly, and this is going to be probability that we will find these bases in this order. So in order to find the length of the fragment, we basically just have to find reciprocal of this number. So we just have to divide 1 by this number, and our answer would be 7,985. This is rounded number. So if GC content wouldn't be equal 50%, but would be 38% instead, we expect that this enzyme would cut our double-stranded DNA every 7,985 base pairs. So you see how GC content would influence fragment size. If every base would be represented in equal amounts of 25%, this going to be a fragment size. But if we will change ratio of the GC to AT, ratio of bases, then as you see, this is how it influences our calculations. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. Share this video with your classmates, and see you in the next video. Goodbye.